I'm Zamprogno Pauline, and I'm currently working at the Organochip Technology Laboratory in Switzerland under the supervision of Professor Olivier Guénin. And our group is specialized in the development of advanced in vitro model called Organochip, and we are particularly focusing on the lung. Our study comes from a simple observation. The standard preclinical model, what with in vitro or in vivo, are not predictable enough to extrapolate conclusion for human. This is the reason why we wanted to develop a relevant in vitro model which could help us to understand better human biology in health and disease, and also predict accurately the effect of new drug on human. In our case, we are working on Organochip. They are microengineering cell-based system which is born of the convergence between macrophagic engineering, biomedical and material science. Thanks to this kind of device, you can really reproduce the cellular composition as well as the microenvironment of a human tissue. Typically, you can mimic the ECM composition, structure, the chemical microenvironment, but also some dynamic parameters such as the breathing motion. In our case, we have developed a lung-on-chip which is based on a biological membrane. The fabrication process here is extremely simple. It only consists of pipetting a drop of collagen and elastin solution on top of the crate, and after a gelation and drying step, we obtain an array of extracellular matrix. We observe that this substrate is really good for cells for grow and proliferate. I think one of the big questions will be the validation of the model. We know that we can integrate a lot of parameters, and actually when you are developing them, you really have to select the one which is important for you, because the number of things that you can reproduce thanks to this kind of system, it's extremely large. So you really have to pick up the parameter which will help you to answer your question. And now I am using this model for IPF on-chip model. So IPF, it's idiopathic pulmonary fibrosis. It's a chronic and severe lung disease, which is characterized by the progressive scarring of the airway. The consequences of this disease is the increase of thickness and stiffness of the tissue, and this is something that we can reproduce in our model. So one of the challenges that we are currently facing on is really about validation and standardization. Basically, we really work with all stakeholders, what with industry or academics, to be able to provide system which is a bit user-friendly, which is adapted for lab equipment and so on. It's really to create enough robust and reliable data to validate our model. This kind of model are a promising tool to reduce and potentially replace animal model. So yeah, this is the hope actually.